My name is Juozas Gordevičius, and I am the founder of VUGENE. It's a bioinformatics company offering multi-omics solutions for research and small and big pharma. So my background is computer science, and I did my PhD in very large databases and data mining. And then I moved into bioinformatics, and I worked in academia for quite a few years. And then I started a bioinformatics company, which is combining the best of what is available in data engineering and bioinformatics to provide services to research in academia as well as to pharma. And we have been very successful so far with VUGENE, working with our clients in the US, for example, with Corewell Health in Michigan, with the Epigenetic Clock Foundation in California, and now we are also making steps into pharma in the US. So multi-omics is a way of looking into the processes that are happening inside the cell at different layers. So we can imagine the DNA as a single layer, then the epigenomic, the DNA modifications and histone modifications as another layer, the transcription that is happening in the cell as one more layer, and then the proteomics, metabolomics as different layers that are happening inside the cell. And then multi-omics is looking into all of those layers, or at least a few of them simultaneously. And what it is special at is we can see how the relationships, how the signal is traveling between different layers, and how that signal can break down or can be changed in disease. And therefore, we can target those relationships between the layers and create new types of drugs and treatment strategies. It is commonly thought that we have a lot of the data, and that's somehow true because a single experiment, a single sample can generate, let's say, 1 terabyte of data. And technically, that's not so much. There are problems and issues that generate more data than that. And so there are cloud systems, there are engineering solutions that can help you to handle all of that data, and we don't consider that a problem anymore. The bigger problem is lack of samples. You can have one sample, you can have an experiment with 100 samples, but it still is not enough if you are measuring millions and millions of different things that are happening across one single cell. And then if you are doing it at a single cell level, then you are having multiple cells. So we need more samples, and that's the biggest challenge that there is. And therefore, the solution, of course, is to integrate with your experiment with other publicly available data, to use AI foundational models to increase the variability, to increase the scope of your data. And that allows to actually gain new insights from quite limited experiments that are limited in size in the number of samples. We are at booth number 1057. We are looking forward to any companies who have multi-omics data and need that data processed, studied, and analyzed in a very fast and top quality fashion. So that's what we are doing. As I said, the size of the data, the computational infrastructure is not anymore a problem for us. So it's the insights that we can take out of this data, that's the value, and we are striving to generate this value to the research community, to pharma community at large.